Welcome, everyone, to today's CDD-sponsored webinar entitled New Frontiers of Drug Discovery, From Machine Learning to Physics. Computational drug discovery approaches are increasingly being applied with the goal of rapidly advancing best-in-class drug candidates into the clinic. Among the front runners in the industry are Reverie Labs, a startup that is applying machine learning to drug optimization, and Silicon Therapeutics, a company pioneering physics-driven drug discovery that was acquired by Roivant Sciences. We are joined in a discussion with Jonah Kallenbach, CEO of Reverie Labs, and Dr. Woody Sherman, Chief Computational Scientist at Roivant, as they share their computational approaches to improve drug design. Jonah co-founded Reverie in 2017 and participated in the Y Combinator program. They have since raised more than $30 million in funding and closed a major partnership with Roche. The company focuses on engineering next-generation kinase inhibitors for oncology by applying cutting-edge computational tools. Woody joined Silicon Therapeutics in March of 2017 from Schrödinger Incorporated, where he was vice president and global head of the application sciences team, helping pharmaceutical companies apply computational chemistry tools to challenging targets. Today's moderator is CDD's own Peter Galick. Peter is a research informatics a senior scientist at Collaborative Drug Discovery, where he develops production quality drug discovery software. In addition to his work at CDD, he teaches data science and machine learning at the School of Data Science of the University of Virginia and at statistics.com. Previously, Peter worked for 20 years as a computational chemist at Novartis. He published on cheminformatics and drug discovery in more than 50 peer-reviewed publications and co-authored 2 books so far. I'll turn this over to Peter in a minute, but first I wanted to invite attendees with questions to be aware that we will be addressing questions following the panel discussion. So please type your question into only the Q&A tab on the Zoom meeting, as we will save all questions for later in the broadcast. So, okay, Peter, gentlemen, feel free to begin.
Okay, thank you for the introduction, Frank. Welcome to Jonah and Woody. Let's just jump in right away. Let's start with you, Woody. Your work at Roivant. Tell us about Roivant and your research there and how you integrate computational tools and what's interesting about the specifics.
Great. Thanks, Peter. Thanks, everybody, for joining. Great to be here. Yeah, as this slide shows, and as was mentioned in the intro, Silicon Therapeutics was recently acquired by Roivant Sciences, so now we're part of this much bigger entity, which is great. So Roivant Sciences as a whole does everything, covers primarily the clinical operations and has been very successful in clinical operations with many successful phase 3 readouts and many before. And now what Silicon brings to the table, which is now Roivant Discovery, is the preclinical capabilities. And from a computational perspective, we've built out a platform primarily based on molecular dynamic simulations, long time scale simulations, accurate calculations with quantum mechanics, and machine learning and AI capabilities, without having a specific tool or specific app, really focusing on understanding the fundamental biology associated with a disease target and developing bespoke tools to solve those particular problems. And so that means, a lot of binding energy calculations, but a lot of stuff going beyond binding, dealing with conformational changes, allostery, agonism, discovery of cryptic binding pockets, understanding protein-protein complexes or induced proximity, ternary structural complexes, which are very important to understand and highly dynamic, going beyond, again, a single static structural representation. We're typically running long time scale simulations on the order of microseconds to see these biological motions of interest. But we also have a suite of machine learning capabilities and tools for doing large scale virtual screening and data sciences work. I think the next slide has a little bit of the details about the company. We have a large supercomputer cluster. Everything that we run is internal. We can run on the cloud as necessary, but most stuff is in our internal cluster. Lots of GPUs on the platform side, quantum mechanics, molecular dynamics, and AI and ML, as I mentioned. And we do have an in-house lab. So we have a combination of about 50-50 experimental and computational folk on the team. We span the spectrum from chip to clinic. We're all in-house right here in our Boston Seaport labs, where we have chemistry, biology, biophysics all under one roof, working with the teams. And as I mentioned, a robust clinical pipeline from Roivant Sciences and all the associated advance, and a very strong preclinical pipeline as well that we're working on. The next slide has a little bit of details about the computational methods that we're developing. We have a bespoke force field. We run quantum mechanics on every small molecule that we're looking at to generate a high quality force field for the simulations. On the simulations front, I've just mentioned a couple of the methods that we've been working on and done some really novel developments in these areas. Weighted ensembles for more exploratory sampling, a metadynamics and extended adaptive biasing force approach for more targeted sampling. We look at small molecule interactions, protein-protein interactions, small alchemical binding free energies, and machine learning, all associated with the various problems at hand. And again, this all relies on having enough computational power to run the simulations at scale to get ahead of the questions that we're asking in the project, so we could actually impact decisions. And so we're typically looking at hundreds of molecules on a daily basis. The cluster's saturated all the time, these simulations are really driving our decisions. And I think on the next final slide that I have here is just some different representations to show that the ultimate output of these simulations is typically not a number, although we do get numbers, and we're very interested in the actual accuracy, the statistical accuracy of the calculations. A lot of the value comes from different ways of communicating the results, different molecular representations, and these are often 3D or even virtual reality based visualizations, because the ultimate goal is to provide the scientists with a deeper understanding of the biological systems of interest, so that they can learn and make more informed decisions to drive our projects forward and overcome key bottlenecks. And so we combine genetic data with structural data to understand mechanism of action and uncover novel binding sites. We decompose energetics into atomic contributions, so we can understand not just what is better or worse, but why is a particular molecule better or worse. A lot having to do with solvent thermodynamics, water thermodynamics, and various different things that come together there. So it's kind of a high-level summary, and I guess we can pass it off now to Jonah.
Mm-hmm. Yes. So Jonah, you're a co-founder of Reverie Labs. In contrast to conventional drug discovery companies, Reverie Labs was founded based on the idea that computation can accelerate drug discovery and is built around this idea. How does this translate into the research being done at Reverie Labs?
Yeah, absolutely. So first of all, I'm really excited to be here. Thank you guys so much for hosting this. Peter, thank you for hosting the panel. And I'm really excited to be speaking alongside Woody, who's a longtime hero of mine and something of a legendary figure in this field. So when we both answer a question, listen a little more carefully to what he has to say. So at Reverie, so we're building a pharma company that's focused on using computational techniques to engineer next generation kinase inhibitors. And so if you go to the next slide, I think, yeah, so just a little bit of background on what kinases actually are. So a kinase is a specific type of enzyme that catalyzes the transfer of a phosphate group from ATP onto a substrate. So all kinases perform this reaction. And as you guys probably know, adenosine or ATP is adenosine triphosphate. So there are 3 phosphate groups hanging off. And the kinase is just taking one of those phosphate groups and sticking it onto something else, usually a protein or a lipid. And it turns out that this reaction is incredibly important in biology, across all kinds of organisms, across all kinds of cellular processes. And the reason is that often these reactions serve as signal carriers and master regulators of important processes. So you can imagine that when kinases are mutated or dysregulated, this often leads to many different types of diseases. And kinases have already proven to be a really fruitful class of drug target. There are already more than 50 FDA-approved kinase inhibitors that are on the market, and cumulatively, these have saved hundreds of thousands of lives. But the problem and the main difficulty with developing kinase inhibitors is that all of those 500 kinase inhibitors or 500 kinases that you have in your human genome are all structurally similar, right? At the very least, they all bind ATP. And so you can imagine that they're going to have a lot of structural features in common. And so designing a drug that just hits one particular kinase without hitting the rest of the kinases and without causing toxic side effects is really difficult. And so that's kind of the central challenge that we have in developing kinase inhibitors, in addition to developing drugs that have all the other drug-like properties that we care about. So if you go to the next slide, so this sorry, I think you went too ahead.
Oh, did we lose it?
Back one more slide.
Yeah, I think we lost it.
Okay.
Do you want to show that?
Oh,
do you have a handy?
It's okay.
All right.
I can just talk through it. It looks like a slide got deleted, but basically the point of that slide is that the structural similarity between kinases is actually a big advantage for a computationally driven company for a few reasons. First of all, it means that the assays are commoditized. So it's really easy for us to generate lots of high quality data against various kinases and be able to drive our programs forward with that data. There's also just lots of pre-existing data out there, both structural data, so crystal structures of protein kinases, as well as historical activity data. And so all of that helps us with building models against kinases. And I'm sure, as we'll discuss today, the quality and quantity of the data is one of the most important things to think about when you're doing any kind of machine learning in any domain, but especially in molecules where often we have much smaller data sets. So on this slide, so there are just a few different elements to our platform, which I've tried to sort of briefly summarize. But in order for computational techniques to have a big impact on drug discovery, you need to both generate the right ideas, you need to put those ideas in the right representation, and then you need to evaluate that representation with a model. And so at Reverie, we use a broad spectrum of techniques. We like to say that we're not dogmatic about any particular technique being right for any particular problem. We really want to let the data and let rigorous validation tell us what techniques are working on what types of problems. But in terms of representation, we'll use everything from a pose of a small molecule in a protein binding site. You can convert that into a voxel grid. You can think of that as a graph. You can think of a small molecule as a smile string, and you can use large scale language modeling. So there's all kinds of different ways to think about how we might featureize a molecule. And I would also just emphasize, potency and selectivity, while incredibly important, are not, they do not a drug make. You still have lots of other important properties that you need to optimize in order to develop a drug that's actually going to be safe and efficacious in human beings. So if you go to the next slide, so yeah, just a quick summary of where we are as a company. So last year we closed a multi-target partnership with Roche and Genentech to work on some different kinases with them. We also have internal programs, including one program that's in lead optimization and another program that we're going to launch soon. And we have a really extraordinary team of scientists here at Reverie, including everything from machine learning and software engineering to computational and medicinal chemistry and biology. So it's a really exciting time to be here. Back to you, Peter. I think you are muted.
Yes, I'm muted. What you said brought back some memories. I worked on the alpelisib compound class during my time at Novartis. So it's always good to see when one's work in early drug discovery leads ultimately to a product on the market. Well, founding a drug company starting from a computational approach to drug discovery is unusual. Jonah, tell us about your path that led to this.
Yeah, so for me, it actually all started when I was a kid with my grandfather, who was a chemistry professor. And since I was a little guy, was kind of steeping me in the world of protein structures and organic chemistry. And I think in college, I studied computer science and a little bit of chemistry. And graduating, I really wanted to work in pharma, and I interviewed at all of these pharma companies. But I found that the computational people were kind of often a side room by themselves, or you would talk to a 20,000 person massive global pharmaceutical company, and they would have 20 CAD scientists who were barely being listened to. And so I think part of the reason for starting Reverie was to build a company where we put computation at the center of drug discovery, and had computational techniques and computational people in our DNA, as opposed to sort of a secluded side room. So yeah, that was kind of the reason that we ended up starting Reverie, and since then we sort of focused in on this area of kinases, where we realized that we had a lot of significant barriers to drug discovery had sort of been lowered.
Okay, so Woody, over the years, you worked in a variety of roles. What influences along the way led to your current role?
In some sense, very similar to Jonah, seeing how things were being done, seeing the potential for computation to make a huge impact, and seeing that sometimes it did make an impact, but it was probably limited due to a lot of organizational and structural, and historical issues having to do with how companies operate. So my focus has always been on the computational side of the equation, physical chemistry and computer science, and worked for a little while in the biologic space during an internship at Biogen, actually, which helped me really see that fantastic research could be done in industry. But there, the challenge was going up not against traditional paradigms, but just the fact that nature can do biologics and antibodies very well, and computation could contribute a bit. But it's really in the small molecule space where I was most attracted in terms of the maximum impact that computation can apply. So that was where things started at Schrödinger, and during my time there, we developed a bunch of great tools, helped a bunch of customers progress projects, and had a lot of successful collaborations. But ultimately, computation was still kind of an add-on, almost like a service where decision makers and projects would ask for help, but computation wasn't necessarily at the center of the decision-making process. And that was really the foundation of Silicon Therapeutics, where we built initially the computational platform and brought the people in, and then started building up our drug discovery projects around that. And one of the key things that I learned at my time at Schrödinger, when we were working with all of the big pharma and small companies, is that drug discovery is hard for many different reasons, and different targets are hard for different reasons, and it's very hard to have a one-size-fits-all solution or a black box, and that's where I think companies like Reverie and ourselves are building out not just a tool, but the expertise and the people and the infrastructure and the software, so that we can develop bespoke workflows to solve very specific problems along the way. And that to us feels like the future of drug discovery. It's not at all different than experimental approaches. There's experimental assays that one might think are just routine, but for every target, there tends to be unique challenges, and that requires experts to get in there and solve those problems. And the same is true with computation. Standard docking, as everybody knows in the field, isn't going to necessarily get you what you need just by running things out of the box. But as you get more expertise and get people who can get their hands into the code and develop specialized workflows to solve specific problems, the quality of the results, the accuracy can go up considerably. And that's really the need to have all of this computational infrastructure in-house.
Yeah, so you already mentioned the silos. At the beginning of my career in the pharma industry, we had groups with molecular modelers, we had cheminformaticians, we had bioinformaticians, and we had a few statisticians as well, all working in silos with often little interaction. And since then, the pharma industry added data scientists, leading to further subdivisions. So this separation is often determined more by organizational structures than actual needs. So Woody, your title at Roivant is Chief Computational Scientist. Does this reflect a shift in how you and Roivant see collaboration within the computational community? And how does collaboration extend to other scientists in your company?
Absolutely, yeah. We do see that we're at the dawn of a new era in computation making an impact in drug discovery. And people have been talking about this for a long time, and there's been a lot of historical limitations, a lot that you hit on, which are the organizational limitations, but there were also limits in terms of true impact, in terms of computational power and the data that's needed to drive a lot of these machine learning models, or even just validate the physics-based models. And so the field has been evolving, and I think we're at a very healthy position right now where computation can really be central in the decision-making process and provide a lot of value. And we're demonstrating that. Others are showing that as well. We were able to advance our small molecule STING agonist program into the clinic with making only a few hundred molecules and really coming late to the game when most of the big players in the field had already been working on STING as a target for years. And we were able to design quite a unique small molecule that's currently in phase 1 clinical trials. And we developed a lot of custom algorithms. The molecule actually dimerizes on itself through a pi-pi stacking interaction in the binding site, but has true small molecule-like properties in solution. You can imagine using standard tools, it's going to be hard to cut it. But with the right people and the right capabilities, we can design algorithms to solve specific problems like that. I think the validation, it's becoming clearer and clearer through a lot of great work in the field, including a lot of great work at pharmaceutical companies and software companies as well. So it does seem like we're at a critical point in the field, and I think we all can appreciate that there's also a lot of hype out there coming from industry and academia, and a lot of people telling the good side of the story. So it's going to be very interesting to see how things pan out and where we go from here. But I think the data is pretty clear that with the right team, meaning not just computational, but the integration with experimentation, and the right organizational structure, and the right tools and capabilities and people, we're going to be seeing an increasingly growing role for computation in drug discovery.
A few hundred compounds to get a compound to the clinic is really impressive. The number of compounds that went into the PI3 kinase inhibitor is probably more in the thousands. I would say probably close to 5,000 easily.
Right.
Jonah, how are the roles of chemists, biologists, and computational scientists at your company defined, and how do they collaborate together?
Yeah, so I think similar to Woody, I agree that the integration of all of these different domains is tremendously important. And we actually view, most of these roles as kind of a spectrum. So, we have, computational chemists at Reverie who are, familiar with and know some ML, and we have ML scientists who are quite proficient in cheminformatics. And it's, we have sort of a spectrum of people on the knowledge, the spectrum of knowledge from chemistry to, machine learning or computer science. People who are sort of all along that spectrum. And so, I think being flexible in terms of the work that people are doing and maximizing the impact that people can have on the projects is really important. And then in the context of our specific projects, we have folks from different backgrounds, including chemistry, biology, and computer science, all working on those projects.
So, Jonah, we heard about the importance of computational techniques in drug discovery today. What technologies do you use at Reverie Labs? Sorry.
Yeah, so I mean, as I kind of alluded to in the beginning, we use a variety of techniques that, as I said, we have a lot of techniques in the quiver, and we try to let the actual results on specific systems tell us which techniques we're going to use. But that includes everything from sort of more conventional physics-based techniques like MD, QM calculations, free energy perturbation calculations, through machine learning models. And I think the machine learning models really shine in areas where you have a good amount of clean data, or you're predicting properties where you have significant historical precedent. So you have for example, basic physicochemical properties like solubility or permeability, where you might have historical data sets that you can draw on in order to predict those properties. But it really spans a pretty wide range in terms of the type of machine learning models and the types of physics-based models that we use.
Data are really very important. At the beginning of my career, data were distributed in Excel spreadsheets by email, and lab notebooks were paper and regularly archived, so they disappeared from your desk and you did not have access to them for quite a while. So what drug discovery software tools have you used over your career, Woody? And how has the need for tools evolved over time and shaped the development of these tools? So Woody, as you worked at Schrödinger and played an active part in the actual development of tools, why don't you give us your take on this at first?
Yeah, so most of the tools that we develop there and a lot of what we do here is in response to specific needs in projects. And so, in some cases, if you're dealing with a novel target where there's no known chemical matter, then data is really not an option, and it comes down to all the physics. And although there is often data, not necessarily on chemical matter, but there's data on the genetics that are associated with the disease, for example. And so we often tap into that genetic data to try to have a better understanding of the disease rationale and how to modulate the disease, and which might not be just a competitive inhibitor, but might be an allosteric regulator, and to understand that allosteric mechanism and the functional role of the particular mutations involves a lot of simulations. And those simulations tend to be, pretty customized for a particular target. But the general infrastructure that we've built, and a lot of the tools that others have as well, starts with quantum mechanical tools, molecular dynamics tools, and informatics-based tools, and then you build up from there. And the key is having flexibility, having access to the source code. I saw one of the Q&A questions already is, do we build everything from scratch or deal with academic code? We always start with what's there. If there is something there that's useful, that's generally a good starting point as opposed to reinventing the wheel. So if there is the appropriate open source code, we start from that, but we typically build a lot around that. And a lot of the stuff we're trying to do, there isn't any open source tools, and then we develop the tools from scratch. But I think an important aspect is, rather than the more historical, monolithic, massive program to do one thing, it is more about having a toolkit, well-structured APIs, typically through Python, so that scientists can solve specific problems. I think there's historically been, people knew that this was important over time, and historically people licensed tools like Pipeline Pilot, which were kind of monolithic programs to make it modular and easy to do certain things. But then that was quite constraining and didn't scale very well, and it was really more for informatics-based stuff. And so I'd say that concept has been around for a while, but making that a reality and being able to solve real problems in real time is challenging and requires a big software team, and it requires a lot of thought on the basic infrastructure, tight collaboration with the high performance computing team. And so that's really the foundation. And then when we talk about specific tools, specific applications, we use aspects of all of these for different problems at different times, but often it is layering on the experimental expertise, often even biophysical data. And I think that's a big area of upcoming value that we're going to see, and that we've already seen internally. And I think the field as a whole will start to realize how we could start to include very specific biophysical data to start to gain additional insights and actually start to drive the simulations in more biologically meaningful ways. And so that's where I think the evolution of the tools have come over time, where it's mostly been historically ligand-based tools, QSAR modeling and similarity screening have been most productive. A lot of the tools like docking have not been as successful because there's been so many gaps in terms of dealing with protein flexibility in waters. And I think the main shift that we're seeing now with GPUs and the power is that we can actually run all atom molecular dynamics simulations at timescales that are relevant for biology. And that does require a lot of computation. It requires a lot of specialization, specialized tools, because we can't just run a simulation and just see what happens. We have to ask specific questions and have very focused analyses, and even the simulations need to be customized toward this particular problem. But that's, I think, where we've seen the field come over time, is from more data and informatics-based approaches to this kind of intermediate regime of structure-based drug design, which was fairly rigid and docking. And I think the promise of that and the impact of that, has become, we've come off the hype curve, quite significantly. But now I think we're starting to see that was the right idea. It's just the tools weren't good enough. The computers weren't powerful enough to actually do meaningful simulations with those structures. And so now we're entering a new paradigm where we can actually run all atom simulations routinely on hundreds of molecules on a daily basis and come up with these much deeper insights and more realistic models.
These are great points. Jonah, what are your thoughts on that?
Yeah, so you were asking about some of the tools that we use in drug discovery, and I certainly agree with Woody about sort of not reinventing the wheel. I think that they're especially within the realm of sort of physics. For us at Reverie, we work with folks like OpenEye. We're not going to write a sort of rocks similarity search algorithm from scratch because there's already a fantastic implementation from the folks at OpenEye. I do think, managing the data is obviously tremendously important. We use CDD Vault for that, which I suppose is why we're here. But I think having the ability to cleanly access your data, with APIs and access a lot of these tools that come from different vendors that may or may not even have a Python API, being able to build sort of clean infrastructure for dealing with all of those different tools is tremendously important.
Oh, I guess you got us there. Okay, I think it's over to Frank now for a poll.
Yeah, we have quite a large audience today. So Charlie is going to run a quick poll. We're going to poll the audience with a question of interest.
All right, everyone. I'm going to set up a first poll here. We are interested in knowing what is the greatest gap in your use of data. So if the attendees could please respond to the poll and make their selections, that would be much appreciated. So yeah, I see some front runners coming in. I'm not sure that we'll be terribly surprised, but we'll give the attendees a little bit of more time to make their selections. All right, seems to have slowed down to a crawl now, so I will end the poll in 3, 2, 1. Thanks, everyone, and here are the results. So I guess the majority, 35%, feel the quality of data is the greatest gap, with quantity and integration coming neck and neck. So I think in some of our preparations, we kind of used our crystal ball and thought this might be the case. Panelists, any comments or ideas on these results?
I think data quality is certainly a huge issue, and especially with the large quantities of public data, even if they're high quality independently, merging those together can produce not high quality data just due to different conditions that are not always represented. So it's not always just the quality, but also just the metadata, the standards around the data. It's hugely challenging and very time consuming to clean up the data sets. Even with professionally curated data sets, there's tons of time that needs to go into that. So I think that makes a lot of sense that's the top one. I think we deal with that routinely. It would be nice if there was higher quality data out there, and it's not always just about the quality of the experiments. It's about, the metadata and how do you combine all this data together in a meaningful way.
Yeah, I totally agree with that. Pick your favorite target. If all of the data in Kemble for that target, was totally clean and generated under identical lab conditions, using an identical protocol, we'd be in a much, much better place than we are as a field. So certainly I agree with that. I also think, for machine learning, we are under sort of fundamental constraints in terms of the quantity of the data. Models can only generalize if they've seen, a sufficiently diverse sort of training set of the property that you're trying to predict. So that's also something that you can't really compromise on, depending on the property that you're trying to predict.
Good. Let me just interact a little bit with you guys. So I want to also let the audience know that we're still taking questions. So make sure you find the Q&A tab at the bottom of the Zoom panel and submit your questions there. We're gathering quite a few at this point, so feel free to do it there. We'll try to answer as many as we can by the end of this afternoon. Back to you, Peter.
Okay. I'll probably blatantly plug 2 publications I have about the comparability of IC50 and KI data. That goes way back a while. Yeah. I think it's better than we may think sometimes. Okay, we just heard how important high quality data are for a successful drug discovery project. The high quality data requires a reliable informatics platform. So what was the decision-making process for choosing the informatics platform at your companies? Who wants to take this one first? Woody?
Sure, I can start. You know, I had worked with CDD in the past. You know, at Schrodinger, we worked with a lot of small companies that had CDD, so we had to do a lot of integration there as well. So the fact that it was, you know, not something entirely new, that it was tested, that there were people and testimonials and all of that was important. Having an API was important as well. You know, not just being able to upload through a standard graphical interface, but something that programmers can work with, allows us to integrate it much more tightly. There's no one program that can do everything, and so that ability to get into the guts of a program and extract the data that you need, or push in the data that you need, and customize it to your needs was really important for us. You know, we evaluated a few different tools, and CDD kind of hit the key points. The deficiencies weren't deal breakers. You know, the people, you all have been easy to work with. You know, you're highly responsive. That's another big plus is just the support team. And you know, Barry's obviously a great guy, has built a great organization, and always has that big smile. So that helped a bit as well.
Jonah.
Yeah, a pretty similar process to what Woody said. I'll say, you know, one unique need that I think we had at Reverie was we needed a system where we could both, you know, easily access the data and serve model predictions, and then sort of upload those model predictions as you know, sort of first-class data into the CDD vault that we use for our projects. And so, you know, having something with a really clean API where we could hook up the models and easily, you know, generate predictions and update models was a key criteria for us.
Mm-hmm. So in both your companies, collaboration between chemists, biologists, and computational scientists is crucial for success. Sometimes project teams also include external collaborators. So Jonah, what is the value of a collaborative system that all team members can use together? Do you think that an electronic lab notebook is critical to this collaborative work?
Yeah, I mean, I think the main value is just, instead of having to constantly be sending Excel files back and forth, and you know, using a data room or something like that, you know, you have a single unified system where the CRO users can log into your account and upload data, and you know, on our end, you know, see exactly what they want them to see. So you know, it just saves your team members a lot of time. Yeah, I think the electronic lab notebooks are certainly helpful. You know, certainly for when it comes to filing IP, it's a really nice thing to have. You know, I wouldn't say that that's sort of the key to the CRO collaboration, but yeah, it's certainly a nice to have.
Okay, Woody.
Yeah, I'd say, you know, getting all the data in one place with a unified view, or at least unified views is super important. And you know, we you know, internally everybody has access to CDD, so everybody's, you know, constantly looking at it. But we, again, through you know, the API, we can extract data, and we built our own web interfaces that are geared toward very specific projects, project dashboards, and specific properties and specific aspects of problems. And so we've basically built out a kind of an ecosystem around CDD, and it's central to have this central repository. And it also helps with just upload from CROs as well, even though they're not looking at our data, just making it easy for people to upload things externally. It just takes out a few steps of human intervention. You know, collaboration is ultimately what's going to, you know, lead to success or failure for all of this. Drug discovery is just way too complicated to have people in silos and have people not get access to all the data. So I think that we all can appreciate now that you know, collaborative tools are central, but how they're implemented and how they're used, you know, I think will be highly variable. And having a flexible tool, again, with a good well-documented API, allows people to add that layer of customization and flexibility needed for their companies.
Yeah, these are good points. So maybe we should take the next poll, Frank.
Absolutely. Charlie.
All right, everyone. So I will launch a new poll for the attendees here. We're interested in which technology breakthrough do you think will have the largest long-term impact for biotechnology? So please, attendees, make your selection from the choices. I don't think anyone's going to be surprised by this. I'm just kind of watching them come in. I get to see them as they're in progress. All right. The votes seem to be slowing to a crawl, so I will end the poll in 3, 2, 1. All right. I'll share the results with everyone here. And a big 60% think the AI/ML technologies is the largest impact breakthrough so far. So I think it has a wide margin here, guys. Any thoughts or comments? Or you know,
it looks like a very biased audience here.
Could be the title of the webinar. I don't know.
So same question to Woody and Jonah. Which technology breakthroughs do you think?
Yeah,
Woody, can you go first?
Yeah, sure. You know, I would have voted on integration of biophysics with molecular dynamic simulations as my pick, but it wasn't on there. I didn't get to anyway. So, you know, I think I think all of these obviously are going to have a huge impact, and in some sense, the robotics and the automation may lead to the data that we need for AI and ML to make that big impact. Cloud computing, obviously, for building these big models is going to be important. But, you know, I think we're all starting to see, you know, impacts of AI and ML. I think we can see the promise. But I think it's also important to, you know, just to remind us all that, you know, we're a very long way off from end-to-end solutions with AI and ML solving drug discovery as a whole. You know, it's still these fairly deep and narrow applications, which can be quite good. And what that means is that you still need a really strong team of drug discovery scientists, and you need to identify specific problems, and you need to build specific AI or ML models that are meaningful for the particular problem at hand, which also requires an understanding, is this data even representative of the stuff that you're trying to predict? So as far as breakthroughs, you know, I would say certainly over the long term, you know, we can all be very confident that AI and ML will be transformative. In the short term, I think there's companies like Reverie and others who are thinking about this, you know, very carefully and choosing, you know, the right target classes where the right kind of data exists, so that AI and ML can play a significant role. And we're starting to see, you know, even, you know, companies getting compounds into the clinic that are, you know, claimed to be through these types of approaches. And it's, I'm sure, not just using AI as a whole to create drugs, but, you know, it's using AI for specific problems and ML for specific problems when the appropriate data exists. And yeah, I think it's going to be a huge part of the future. I think we're starting to see that play out. I'm sure there'll be, you know, we're going to go through that hype cycle. So it's going to be overhyped for a while, and then it's going to, you know, it's going to come down, and there's going to be some leaders that emerge who have figured out how to, you know, use it appropriately and create value, you know, when it makes sense, and figure out how to augment the cases when it doesn't make sense with either more data or experimental approaches.
Yeah, good point, Jonah.
I totally agree with Woody. You know, I think AI and ML in a vacuum, you know, can't really do much for us. It's really through the integration with, you know, how we generate that data, how we process that data, how we train models, which requires cloud computing. Even, you know, in our case, requires cell engineering to, you know, generate the types of cell lines that we use to generate a lot of really critical data for our programs. So it is really an integration between all of these things. And I also just briefly, you know, I totally agree with what he was saying about kind of where we are in the development of these technologies, which is, you know, it's still very, very early days. You know, 99% of pharma R&D is still done in a way that does not involve machine learning at all, and probably doesn't even involve, you know, sort of physics-based computation. So we have a really, really long way to go before it's, you know, 99% of pharma R&D is using these techniques. So it's certainly really early days in these technologies, and in terms of seeing, where they work, how to, make them as applicable and accurate as we possibly can.
Yeah, that's a good point. We could probably do a single webinar just talking about how slow sometimes big pharma is in changing. So what are the cultural challenges to the decisions you've made to move forward with technology? Jonah, give us your insights.
Yeah, so I mean, I think that in some ways is actually the hardest problem that we face as a computational drug discovery company is, how we make decisions. Having great models doesn't mean anything if you're not actually using those models to make good decisions in your drug discovery programs. And the industry as a whole is very used to decision-making processes working a certain way that's, largely centered around human intuition. And introducing changes to that framework is really difficult. So, I think for us, encouraging just open collaboration between chemists and computational scientists has been really critical there. I think you need to make sure that people are staying up to date with, which models are working and which models are not working. And, this requires folks on the computational side who are willing to be, sort of totally transparent and humble about, what's working and what's not working and when it might work. And then, open-minded chemists and biologists who are willing to say, hey, it does look like there's, rigorous validation of these techniques. And so we should be using them, in these different contexts.
Yeah, good point. Woody.
Yeah, and just to continue along that, the cross-functional learning is so key. Being able to understand, as Jonah said, what models are working and what are not working. But it's really a continuum, and as we know, all models are wrong. Some are useful, and if we work together with the experimental team, we will be able to build more useful models, and we will be able to use the models in the appropriate ways more frequently. But getting the mindset, because a lot of people are used to working with computers where you just get the answer. You do math on a computer, you get the answer, and it's the answer. And the models that we're building are obviously very complicated, and biology's very complicated, and there's never going to be a one-to-one match. And so, one can run a calculation that's right 50% of the time, and depending on how hard that problem is, 50% might be an awful result, and you might as well flip a coin. But if the baseline is 1% of the time you're getting the right prediction, then 50% is a tremendous improvement. And understanding what that baseline is, first of all, is very hard. It means you kind of have to go through historical data or talk to people or something to figure out, where the baseline is. And then pulling the people together to understand, that these models, when used over and over, will provide value. They will get you to the clinic faster. For example, in our STING program, we have a very nice plot that shows the distribution of the free energy predictions of good and bad compounds, and there's clearly a separation, but there's overlap, and that means on any given compound, any given day that somebody makes, the simulations could be wrong. And to be able to understand that and learn from that, and also present the data in a way that's more than just numbers. If it's just a number saying this molecule is good and this molecule is bad, you don't really need much collaboration. You just kind of need somebody to make a molecule. When you start getting deeper into the insights that are coming from why this molecule is good and why this molecule is bad, and what atoms are contributing, and what's the confidence on the predictions, and what data went into it, now you could start to have a more sophisticated conversation and ultimately get to better decisions that are not just going to be the computer's decision, but is going to use the computational information in a more useful way. And that's where I showed the figures at the beginning, the data representation, and I just showed structural representation, but there's obviously data plotting representations and ML representations and all kinds of other representations that we need to think about. Exposing those in a way that's meaningful to the decision makers is ultimately the key challenge. Even if the calculations are good, if they're not exposed, and having a calculation that's mediocre but presented in an intuitive way is probably going to have a higher impact than an extremely accurate calculation that's just presented as a number. And figuring out what that high value representation is to communicate requires a lot of cross-functional learning and discussion and exchanging of vocabulary and understanding the fundamental challenges, not just in the computational world, but in the experimental world as well.
Yeah.
Fantastic. It looks like we're running out of time, and I want to thank all three of you. We're going to go to questions. Charlie's been looking at various questions, and I hope we have time to address as many as we can at this point. So Charlie.
Yeah, so okay, so first of all, too many questions to answer in the remaining time, so thank you everyone. I picked some from the list from early in the webinar, middle and end, so forgive me if it seems like maybe we're backtracking a bit, but I thought we would give all parts of this some attention. So one of the questions is, we know classical force fields aren't correct and quantum mechanics simulations are very expensive. How are you guys balancing this speed versus accuracy when working with or creating your computational data?
I could take a stab at that. You know, it kind of comes down to that idea of bespoke workflows to solve specific problems. And so anytime there's bond making and breaking, you know, we have to deal with quantum mechanics. You know, when we use quantum mechanics for, directly for things like pKa and tautomer predictions, for example. We actually do that coupled with machine learning. It's a great intersection between physics and machine learning there. You know, the force field, as you mentioned, we use a bespoke approach, meaning that we parameterize every molecule on the fly using quantum mechanics as opposed to having lookup tables. So that's a results in a huge improvement for complicated drug molecules where the lookup table is just insufficient. And so the force field actually does extremely well then when you're parameterized based on the molecules for internal energetics, but you still miss the kind of the polarization effects and the more exotic interactions, pi stacking interactions and stuff like that. One thing that we've done to improve the force field further is adding off-center charges, basically, which can represent lone pairs, for example, and sigma holes and stuff like that. So to get these kind of non-spherical charge distributions. And so that gets a step closer to dealing with, the real nature of the potential. But then we also just add in quantum mechanics as needed for specific problems. So for example, in that STING agonist program that I mentioned that got to the clinic, one of the key parts is that this small molecule that's a small molecule in solution dimerizes on itself in the context of the binding site, and it dimerizes through a pi-pi stacking interaction. And the molecular mechanics based free energy calculations just weren't differentiating the various subtle kind of electron donor the electron pushing and pulling effects going on in this, fused aromatic ring system stacking on itself. And it was more complicated than typically just interacting with a protein, because you're in a protein, at least you have a fixed residue that you're interacting with, and now you're changing both sides of the equation. So we had to develop a method, in very short order in order to impact the project, and we were able to do that. And so we ran our free energy calculations, and we augmented those binding free energy calculations with this pi-pi stacking contribution. And so I think just adding it in intelligent ways to solve specific problems is where we're at now, because as you said, it's just too expensive to run real quantum mechanics on an entire biologically sized system.
Yeah, I would just add, in the context, at least of small molecule calculations in vacuo, QM is expensive, but it's not that expensive. And cloud computing allows us to launch huge amounts of compute, massive clusters of GPU from scratch. And so for solving certain important QM problems, you can just do that. And then maybe as Woody was alluding to, I think it's also a ripe space where you can apply machine learning to approximate results from QM calculations, and get 2 or 3 orders of magnitude in speed up, and not lose that much accuracy as long as you're sort of in domain for your training set. And so there have been a bunch of papers that have demonstrated those kinds of results. There's the Schnet family of models, as well as people have demonstrated this with MPNNs as well. So it's definitely a cool area where you can sort of build your own training set. You can even use active learning to figure out what molecules you want to use, and then you can train a machine learning model to predict the property you care about.
Excellent. All right, thank you. So another question, kind of a two-part from two different attendees that I seem similar. How do you deal with resistance to drugs in the clinical setting, patients becoming resistant, having to be moved to another therapy? And are there recent or any machine learning techniques being employed to solve acquired resistance of drugs?
Great,
Yeah,
why don't you take that, Jonah?
Yeah, I was gonna say that's definitely something we think about quite a bit in the kinase domain, and the reason is that it's what happens to lots of kinase inhibitors in the clinic is that patients develop resistance mutations, and so if you look at sort of the spectrum of companies out there working on kinase inhibitors right now, lots of them are basically working on solving these types of questions where they say, okay, all these different resistance mutants are cropping up, and as we get better and better treatments and cancer patients are living longer and longer, these problems are just going to sort of multiply. And so that's honestly part of the motivation for why you would want to build a company like Reverie, is to be able to solve these problems quickly and efficiently, because it's going to keep happening. And it's, you have to, we design a drug, the cancer mutates, and we have to design a new drug that solves that problem. And so it's kind of this constant arms race between how quickly can we do kinase drug discovery and how quickly are these cancers mutating. On the other hand, the other thing I would say is that I think there's a more fundamental solution to this problem, which is kind of taking an approach like that the pharma industry used to tackle problems like HIV, which is to target multiple mechanisms at the same time, and thereby circumvent the ability for any one of those resistance mechanisms to escape the treatment. And so the problem is, historically, a lot of cancer drugs have been very toxic, and so you can't necessarily give a cocktail of 3 or 4 targeted therapies or chemotherapies, in part because a lot of these drugs are really non-specific. And so, again, part of what we're trying to do at Reverie is develop these really specific kinase inhibitors that could theoretically be dosed as part of a combination regimen with other drugs. And by virtue of being, really selective kinase inhibitors, you might be able to actually, give a set of, hit a set of different mechanisms at the same time, which, currently you can't do because a lot of these drugs are just not very well tolerated.
Perfect. Thank you so much. All right. So I'm going to do 2 more questions because of the time that we have left. The first one is going to involve your ability to polish off your crystal ball and tell us what you think will be the biggest bottleneck for the future of machine learning-based drug discovery.
My feeling is just high quality data.
Yeah, I think that's right. Narrowly looking at sort of machine learning, I think there's also kind of the larger bottleneck of drug discovery in general, which is sort of finding the right targets. And I think that's an area where there are a lot of potential contributions to be made from AI and ML, and companies out there have only sort of scratched the surface of what can be done there.
Excellent. All right, everyone. So we've talked about bottlenecks and breakthroughs and gaps. So I want to end here, hopefully on a positive note. For those out there listening who have an aspiration to join in this new frontier of drug discovery, this computational chemistry world, what do you suggest? How do they get involved? How do they join your party?
Yeah, so for me personally, I mean, anybody should reach out on LinkedIn. I should be easy to find and happy to chat further and point people in different directions. I do hope that there's becomes more, there's no major in university for molecular simulations or any of this type of stuff that we're doing. There's all these great Coursera courses on machine learning, but very few on these approaches applied to drug discovery, molecular simulations, and all of this type of stuff. So I'm hoping that we're going to start seeing this emerge at universities. Universities, some have some of these little one-off classes or lectures around this general theme. But I think that's ultimately going to be what we need is to be educating the next generation of people, and that even chemists and biologists and everybody else coming up through the academic system will have got a flavor of the fundamentals of these approaches, molecular simulations, quantum mechanics, molecular dynamics, and machine learning applications. Because once people understand those fundamentals, they don't need to be professionals in that area, but they will be able to have the conversation, have that common language, which is ultimately going to lead to more successes and more involvement, more opportunities. In the meantime, I think people, probably you got to go get your PhD in this field, or at least, talk to a lot of people. It's still quite complicated, to get into the weeds and really understand things and move the paradigm forward. And there's a lot of experts out there. Go to conferences, read papers, read books. There's a lot of good books by very reputable people with backgrounds in the right areas to see how these computational approaches can make an impact in drug discovery. And mostly, reach out to individuals, read a paper, talk to the authors, and do the basic science work to get into the details. And there's so many facets of molecular simulations and machine learning applied to drug discovery. So it's kind of an endless learning experience. We're constantly going through that same learning experience as well. So there's no unified reference to get it all, but I'd say just start asking questions and connect to people.
Yeah, we're over time here, so I'll keep it brief, but I'll just say, I agree with Woody that this field is exploding, and every day, you're seeing new open source software packages, new papers, new companies that are working on this stuff. So it's a really exciting time to get involved in this field. And you're always welcome to reach out to me, or reach out to contact@reverielabs.com, and we'd be happy to chat further. And also, of course, we're hiring like crazy over here. So if you have the background in machine learning or in computational chemistry, we're always excited to chat.
Wow, good stuff, guys. We could probably go another hour, but I know we can't. Would like to thank Jonah and Woody for a great conversation today, and Peter for chairing this event, Charlie for all your work in this too. But I'll do my little commercial. So lots of things are happening. We're starting to get out there in the world again, which is awesome. So collaborative drug discoveries at doing talks and exhibitions, and this is just September, so things are happening. And if you are inclined to go to any of these, some are virtual, some are combination virtual and on-site, reach out to us, find us, and go to our website. Take a look at collaborativedrug.com. Follow us on LinkedIn, and that's all I wanted to say for today. But this is just the beginning of a lot of good interactions with scientists again for CDD. Thank goodness. So thank you everyone for coming again, Jonah and Woody, for your work today. It's been awesome. And thank you everyone. Have a great day.
Thank you so much, Frank.
Thanks a lot. Yep, and thanks Peter as well. All right,
Thank you.
take care, everybody.
Thanks.
Goodbye.
Bye.
Bye. Bye-bye, everyone.